Alright, so I thought I'd make a quick video about this new SARS-CoV-2 variant that they are calling Variant of Concern 2020-12/01. And so oftentimes seen as VOC 2020-12/01. So this is that new strain that's been in the news in southeast England that seems to be more contagious. And so I wanted to go through this brief here, and so I've taken some of the figures and so forth from it right here, and so I'll talk about that. So in this brief, they use several different names for it. So they use this, which I don't know what that stands for. This was the old name for it, so Variant Under Investigation 2020-12/01. This is now the new and preferred name for it, so Variant of Concern, or VOC 2020-12/01. Alright, so this variant has some mutations in it that are found most often, but the ones that they seem to be most worried about are these ones I have circled in red here. So they all take place in the spike protein. If you've watched other videos I've made about SARS-CoV-2 or coronavirus or COVID-19 or whatever you want to call it, we know that the spike protein is what allows the virus to bind to the human ACE2 receptor in the lungs and gain access into the cells. And so these three mutations seem to be important for making this virus more virulent, more contagious. And so the first one, which is kind of the most important of them, is this asparagine 501 tyrosine mutation. So these are the bases that are mutated in the genome, and this is the resulting change in amino acids. And so they have up here from that paper mutation N501Y, which is asparagine. It's asparagine to tyrosine, and it is this base that is changing. So here it says A to T, but U is the same as T in RNA. So it's this A being changed to this U, so it's going from asparagine to tyrosine. Is one of the 6 key contact residues with the receptor binding domain, RBD, that has been identified as increasing binding affinity to human ACE2. And so I have the structure of that here, and we can actually see, so in yellow here is the human ACE2 receptor, and up here is the SARS-CoV-2 spike protein, and we can actually zoom in here to where this SARS-CoV-2 spike protein is making contact here with the human ACE2 receptor, the human ACE2 here. So this doesn't show the mutation. This is asparagine. I have kind of an overlap going on here, but this pinkish color and this bluish color are the same thing, and this is the asparagine residue here. So this is being turned into a tyrosine. So like I said, you can see here, this is right at the interface where the SARS-CoV-2 spike protein here in the salmon-like color is binding here to the human ACE2 in yellow. So that asparagine is being mutated into a tyrosine. So asparagine has a side chain that looks like this. So it has this double bond oxygen and then this nitrogen, where tyrosine has one that has this this large aromatic ring with this phenolic OH group here, so this should be aromatic. And so this seems to increase the binding affinity of the SARS-CoV-2 spike protein to the human ACE2 here. And what that would do is, it would allow the virus to gain or to bind tighter or bind more often and therefore gain entry more often, but it would also help to sort of bind to it and prevent anything else from binding to it. And so those two things would allow the virus to be more contagious and to actually enter the cells more. So this other mutation here, this proline 681 histidine mutation, this actually occurs, so it says up here, is immediately adjacent to the furin cleavage site, a known location of biological significance. And I've talked about the furin cleavage site and the cleavage of the spike protein and how that occurs in the virus gaining entrance to the cells in previous videos. But just sort of quickly, what happens is, so I'll actually show it here. And so the furin binding site, I actually have, so it actually didn't show up in the structure. So we see up here in the sequence there's this gap here, and that would occur in a loop connecting these two sections here. But that part of the protein was too disordered, so it doesn't maintain a structure. And so that wasn't able to resolve in the structure that I got from this on Protein Databank. But there would be a loop going between here, and that is where another enzyme, a human enzyme, it's actually an enzyme on the surface of the human cell that would come and cut that. And then what would happen is the top of this protein would sort of come off. So it would sort of separate, it would cleave from this, and there would be a large conformational change that happens in the other part that would sort of reach over and grab onto the, it would it's kind of compacted like this, and what would happen is this part sort of moves outwards so that you end up having something more elongated, and that reaches out to the cell membrane of the human cell and allows the virus to sort of gain access into the cell. And so this mutation adjacent to that furin cleavage site is also seems to be important. And then the other one of interest here is this deletion of a histidine and a valine at locations 69 and 70. And so we can actually see that on here. So I have that down here in yellow. So this is the histidine and the valine that would be being deleted. And so these would be deleted, and it would just shorten the actual protein or the amino acid sequence, the polypeptide, by 2 amino acids. Nothing is being replaced there. It's just a deletion. And so what's interesting about that is it seems to change the in the actual viral genome, which is an mRNA, it actually seems to change the binding of the PCR or protein or polymerase chain reaction probes to the viral genome. Up here it just says has also occurred a number of times in association with other receptor binding domain changes. But we will look at a little bit more of the clinical sort of data down here. So this is looking at the ratio of of this growth rate here for this new variant compared to the old variant. And essentially, what you should see on here is this purple line is if the growth rate is well, if there's essentially no growth rate. If it's below the purple line, then there is a decreasing rate. Above that purple line is a growth rate. It's similar to that if you looked into it, that R naught, which is essentially the number of people on average that an infected person will pass the virus to, maybe in some given amount of time. So if R naught is greater than 1, then we know we have growth, because every one person will on average pass it to more than one person. Whereas if R naught is less than 1, then on average that person may pass it. So if it was, say, 0.5, you could say there was a 50% chance that they would pass it to another person. And so this is kind of doing the same thing, where it's using this equation, or this formula here, where it's looking at the number of viruses with that sequence found this week, and dividing it by the number of people or the number of cases of it last week, and getting this RT here. And then to get this, they do this for so T1 and T minus 1 for the new variant. And then they also do it for the not new variant. And then they take those two RTs, so the RT, we'll say, comma, new, and maybe divide it by the RT, comma, old. And that is how they actually get this here. So if this RT is higher for the new than it is for the old, so that would meaning that the new one is more virulent, then they can actually divide that by the RT for the old, and see, sort of get an idea of how much more virulent it is than the old one. And so up here, if this was equal to 1, then we would say, well, it's not more virulent, it's exactly the same, because the virulence for the new divided by the virulence for the old was equal to 1. Now, if it is greater than 1, then we would say, well, this is a larger number than this, because this was larger than 1. And that's exactly what they find up here. So the average up here, this blue line here, is above 1, and so they can say that this is actually more contagious. Its growth rate is greater than the growth rate of the non-new variant. And so, as I was saying before, to actually test these things, and I guess if that was what you were mostly interested in, that what I just said was sort of the crux of this, I'm just going to go a little bit more in technical detail here if you're interested in that. So what they do to test this is actually use this thing called real-time PCR, often called RT-PCR, where PCR stands for polymerase chain reaction. This is sometimes called qPCR for quantitative PCR. And I guess PCR, to give an idea of that, I have this down here. So you take your DNA, so in this case they may, in the case of this virus, since it's actually an RNA virus, they might actually make a cDNA from that mRNA, and then they can do PCR on that. But what essentially it is you denature the DNA, which is essentially increasing the temperature so that each strand separates from each other. Then this annealing step, these primers, these RNA primers come in, and that allows a DNA polymerase to actually come in and add new DNA on here, so that now you've doubled your strands of DNA on the first cycle, and then you just keep doing more cycles, and it keeps doubling the amount of DNA that you have. And so this is a pretty standard technique in cell biology and biochemistry labs to increase the amount of DNA that you have. It increases your DNA concentration. And so for quantitative or real-time PCR, they do this same thing, except they use this probe here. This probe is the primer that they use, and so this comes in. It's quenched because there is this fluorescent dye on here, and this other molecule bound to it that actually quenches that dye. So we have light coming in here, it hits the dye, and then when that light re-emits, instead of coming out so that it can be measured, it's actually quenched by this nearby other molecule here. That's sort of a quantum mechanical thing that I'm not going to get into about how that actually works. But anyway, it quenches it. But then we have it come in here, and then when the DNA actually is actually reproduced, it comes in and actually breaks this part off, the dye off, so that it moves away from the quencher, so that it's no longer quenched. And now while you're shining light on it, it actually hits that dye and then comes back off, and then you can measure it. And so you can measure the increase in fluorescence in your solution as you are going through this PCR process here. So there's this other fluorescent dye based where it's a free fluorescent dye, and that just sort of intercalates. But the one that they were using for this stuff was actually this, I forget exactly what it was called. I think it was a thermal Fisher kit that they were using, though. We can probably look that up on here. Yeah, thermal Fisher. It was called TAC Path. That is what they were using. They were probing for the open reading frame 1AB, and the spike protein on the virus genome. So we can see over here the nucleoprotein is the N. The S is the spike protein. Then we have these open reading frames here, and so they were probing for this right here, and they were probing for this right here. And interestingly, what happens is that deletion that they get at this HV deletion at 69 and 70 makes it so that this which one was it? I think it was the S. Yeah, the S. They can no longer detect when they do real-time PCR. The DNA probe doesn't actually bind to the genome, the S protein DNA. It stops binding to it. The DNA probe can't bind to it, and so they can't actually see. This process going on. They can't see the increasing concentration of the DNA through PCR, and so they call that the S dropout here. And what they found is is that the deletion in this new variant, they're finding an increased amount of that in the proportion of the virus that they're finding. And so you can see that they're finding more S dropout, and that is going up correlatively with this B.1.1.7, which, as I said, is the same as this variant of concern. It's just another name for it. And in blue here are other variants that are not this new variant of concern that have this deletion in it, and you can see that that is actually not increasing, but this new variant having that deletion is increasing at the same time as that variant is increasing. And so they actually have a table down here for that, and percent new variant with this deletion here. So back in October here, October 12th, it was only 5%, and now here, well, at the end of November, it was up to 96%, and you can see how that would be, where if we take all of the S dropout here, so everything that has the S dropout, that is, so that's the whole population of S dropout, and then the percentage of that is the new variant, which would be most of it, so only a small amount here would be represented by other variants that have this deletion. And so 96% of this at the end of November was the new variant. Yeah, so anyway, this is essentially all evidence showing that this new variant, as I said, this was sort of the thrust of the argument here, the crux of the argument, that the new variant is in fact more contagious, more virulent. Its growth in the population is higher than the growth of the old version, the non new variant of the virus. And so anyway, I just thought that would be something interesting, or maybe useful to explain. I think a lot of times, if you're maybe a little bit more literate in science, you read or listen to news stories talking about this, and it's just kind of like they're not giving me that much information, and so I thought maybe I could give a little higher level of a description of this. Anyway, I will see you in another video.